Hey guys, so I wanted to cover chapter 13 in this video. With chapter 13, it's a really big chapter. Like I was telling y'all, chapter 12 before was kind of a little one. Chapter 13 covers a lot of content. It's actually, if we were in our principles class instead of our intro class, this is four different chapters from the textbook, all squished together in one chapter for 1012, which is kind of crazy. So anyway, I think I'll probably chop this video up into the first half, prokaryotes, and then I'll do all the eukaryotic stuff in a separate video. So let's go ahead and get started. So we are going to start off with the prokaryotes, and those were the first guys. So I'm sure we probably talked about this in 1010 if you took 1010 first, but if we didn't, the Earth is really, really old. It's like 4.6 billion years old, and about 4 billion years ago, all the prokaryotes came on the planet and they evolved. And we could talk about different conversations on cell theory and how they came to be. But the first celled organisms came on scene and evolved. So from them, those are all the prokaryotes. Eventually, we evolved the eukaryotes. So the prokaryotes were first, and they were on the planet way before the eukaryotes. So they have evolved and adapted to all the crazy environments where you could think something wouldn't live. These guys live there. Prokaryotes live there. So they're really wild, really crazy. They live in all sorts of really neat environments. And then the eukaryotic cells came later. When we talked about the difference between prokaryotes and eukaryotes, they came later, and that's going to be us and the protists and fungi and all of us. We are all eukaryotes because we have membrane-bound organelles, right? We've got a nucleus in our cell, where the other guys don't have a nucleus. We'll talk about the parts that the prokaryotes have in their cells. But huge diversity. Prokaryotes and eukaryotes are literally everything. The whole tree of life with the three domains that we talked about before, that's everybody. And we've got some imagery here of the different groups. Come on, PowerPoint, work with me. There we go. Speaking of the different types of little bitty guys that you can't see, um, it's always important to use good hygiene, and I like to start off prokaryote chapter every time with this image. So this is from Tasha Sturm, and she was working in a lab and also has children. And her child, the story goes, her child was outside playing, doing things that kids do, and came inside, and she was like, "Here, put your hand on this giant agarose plate." It's just like the plates that we use in regular micro. It's just a bit bigger. So the kid comes in right on to the agarose plate, the agar, and she just sticks it in the incubator, pops in the incubator. And this is all the stuff that grew on it, which I think is just, it's so wild and, really gross to see all of this stuff because you've got multiple different colors. You got yellow stuff, you got orange stuff, you got bright white stuff and ivory colored stuff. You have all the different things. You've got some fungi on here, you've got bacteria for sure, all sorts of stuff. And if you look too, this really big guy in the bottom right hand corner, this guy, I don't know what it is, but whatever it is putting out antimicrobial chemicals out there, and it is killing off everything next to it. It's called the zone of inhibition. Let me see if I can get my little pen working. It's called the zone of inhibition. And so all of this stuff right here, all of this is because it's killing everything. So whatever it's releasing, whether it's antibiotics or antifungals, something, it's releasing some sort of chemical to kill off its competition so it can have most of the resources for itself, which is really interesting and something that I hope um, more scientists are looking at because there are still so many chemicals out there that we have not discovered coming from prokaryotes that we could possibly use selfishly just for ourselves for medicinal reasons. It's just one of the many reasons why prokaryotes are super cool and really important. All right, on to the diversity. They're wild. They're everywhere. Like I was telling you about in the beginning, uh, the Earth is 4.6 billion years old, right? And at the time, it was pretty toxic. There were toxic gases out there. It was, a boiling hot rock with lots of funky water and storms, and the oxygen levels were not the same. So it was a lot of anoxic environments out there, which just means low oxygen. Anytime you see A in front of something, it's just going to mean without. And so we had the prokaryotes that came on the scene, evolved on the scene, and most of those guys were anaerobic. They evolved their metabolism not needing oxygen in order to do the things they need to do for cellular respiration. They didn't have to have it, which is really cool. And then out of those groups, another new group of prokaryotes evolved, and those guys were phototrophs. So these are similar to like we learned about in 1010 if you took that class. They are undergoing photosynthesis. These guys are undergoing photosynthesis. And so on the byproduct of photosynthesis, right, those guys are emitting out oxygen as their byproduct, just like plants do. We are the opposite. We're going to exhale out CO2, but these guys exhale out oxygen. So when all these photosynthetic prokaryotes were coming on the scene, they terraformed the earth and allowed the oxygen levels to get much higher than they were back then. And it's all because of these photosynthetic phototrophs, the very first prokaryotes that were using light and harvesting that energy to undergo its metabolism. And you can see them in this little stream here. These used to be called blue-green algae. Now we call them cyanobacteria. But all of this green stuff in this hot spring right here, that's some photosynthetic bacteria, or it could be archaea too in here. So they are really cool. They're really diverse. They are, again, extreme ophiles. They live in all the extremes. Any wild, crazy places that you think like nothing lives there. It does. These guys do. The prokaryotes do. So when I say prokaryotes, it could be bacteria or it could be archaea. They're two different domains, but together we group them as prokaryotes. So in these extremophiles, we can see lots of these at Yellowstone National Park. So I put in a little picture of the trip I took with my family when COVID hit. We decided to isolate, and we rented an RV that we pulled behind. It was a trailer. We rented a little trailer that we pulled behind the SUV, and my husband and I and our three kids piled into the car with our two dogs, a Rottweiler and a pit bull, and our two cats, all in our SUV. It was wild, crazy times, but we got to see lots of things. So we stayed in our car and in the trailer, and we hit the road and got to see some really cool places. And Yellowstone was one of those. So if you haven't been, I really do try and encourage you to travel and see the things, especially now, more important than ever, to please see your national parks if you can, in the level and pristine beauty that they're at. But Yellowstone is especially cool because it's a caldera. It's a volcanic basin environment. And so all of these pools are super duper hot, like boil you alive hot, and there will be nothing left of you hot. And so you can look inside of them though, and it looks like really pretty and turquoise water, and it looks so inviting, but don't get in. And then on the edges of this guy are these rainbow colors. So you can see orange, and then there's some deeper reddish ones. Those are all bacteria and archaea that live and thrive in those extreme temperatures. It's so cool to see. So there's lots of different hot springs in the Yellowstone area. This specific video here, if you download the PowerPoint and click on the link, is about Grand Prismatic Spring, which is where we're at in this picture. It's a really cool spring inside of the national park, which is our very first national park, is Yellowstone. So if you get the chance to travel, even on a shoestring budget, because that was a shoestring budget to fit us all in that SUV and and sleep in the trailer, please do. Please see it, check it out now more than ever. Go see the parks if you can. Extremophiles and their preferred conditions. We just talked about the ones that lived in really hot, insanely hot springs coming out of those volcanic calderas. So those guys that like really warm temperatures, really hot temperatures, we got thermophiles and we got hyperthermophiles, and you can see the temperature variation for our thermophiles. They like it warm. Hyperthermophiles like it insanely warm, insanely warm. And so those guys live and have grown and evolved in those zones. They're not going to live in your basic tap water, coming out of the faucet temperatures. They need those hot temperatures. That's where they live and thrive. Then we've got other types of extremes. We've got the acidophiles, so they'll live in very low pH places. So there's a type of bacteria that can live in your stomach. It's called Helicobacter, and that's the genus anyway. I think it's Helicobacter pylori. But anyway, they live in your stomach acid, and they can live and thrive in there, and they can cause ulcers. And so we used to think way back in the day, like, oh, you've got a stomach ulcer. It's just because you're stressed. It's just stress. It's not stress. It's a bacteria that's living in there in your, pH of 1 stomach acid. So now we know what that is and what causes it. And we can give you some antibiotics and some Pepsin and send you on your way and treat those. But they can live and thrive in very low pH, like pH of 1, pH of 2. Then on the opposite spectrum, high pHs, alkophiles, pH 13, 14. They can thrive there too. If they like super cold temperatures. Like you would think the polar ice caps don't have any life in them. There, that you would be wrong. You would be wrong. There is stuff living in there. So the psychrophiles like really cold temps, like really cold temps. And then we have halophiles. Those things like it's salty. They like it salty. So if you see Great Salt Lake in Utah, any other, the Dead Sea, places like that, that's where you're going to see halophiles. So if you see the prefix halo, it means salty. And then if it's not salt, we're talking about other types of things dissolved in a solution, osmophiles. So for instance, high sugar concentration could also be something that something has evolved to grow into and love. Most of the time, they things do not thrive in salty conditions. And this is one of the ways that we used to and still do today cure our meats is with salt, heavy doses of salt. So usually those high concentrations of things will kill off most other stuff that would harm us. But these guys, some of these guys are specifically evolved into all of these different conditions right here. All right, microbial mats. These guys can form biofilms that turn into microbial mats. They can live in all sorts of different places, like we talked about. Sometimes they could be phototrophs, and that is where they're going to use light as their energy source. So that's these guys over here. If you had your 1010 lecture, you know what I'm talking about. Um, so they're going to use light instead of chemicals. But some of these guys, and this specific one is talking about growing on a hydrothermal vent, which is at the bottom of the ocean, and those guys aren't getting any light down there. So they are functioning off of chemotrophs, not off the light, because there's no light way down there. They are going to be chemotrophs on there. And so those guys can build up mats. They'll build, make this biofilm. And then sometimes these biofilms stay for so long that they eventually will fossilize, and those guys turns into stromatolites. So we've got these. This is a picture from Glacier National Park, which is my absolute favorite national park, is Glacier out in Montana. And it's fossilized stromatolites, which are nothing but a fossilized microbial mat, which is really neat. I'm not going to ask you about steps of biofilm development, so please don't stress out about this image that I added into the PowerPoints, because they're not in the textbook. But I just wanted to show you like what a biofilm was. Like this is where the little guys find a place to live. And every time I talk about biofilms, it makes me think about the plaque on our teeth. That's a biofilm. So like, I did brush my teeth today. But these are the little bacteria that live all in our mouth. We've got at least three different species, at least living in here in a little biome. And you scrub that off, I hope, every day. Hopefully twice a day. If you're following good hygiene practices, you're brushing that off twice a day. But here's the beginning. They find the place they like and they think this is kind of great. And then once they really figure out that they like it, permanent attachment, and then they're going to grow and express all their different little genes and they're going to do what they want to do. And then they will be as mature as they want to possibly be. And the cool thing about this, or maybe the uncool thing about this, is when this biofilm forms, if you look at this little yellow stuff right here on the edge, the yellow stuff is called a polysaccharide layer. It's kind of like I love food. I'm always going to think about food. So I think of it like an M&M, like the candy shell on the outside of the chocolate for your M&M. That's kind of what this is, a polysaccharide, sugary, crusty shell. And it's so if you get a biofilm, say on like a chemo port, for example, and it gets an infection, it is hard to treat those because of a biofilm. It's hard to get the medicine to penetrate through this and into the bacteria that are in the heart of that biofilm. Sometimes the bacteria in the heart of the biofilm have shut down their metabolism. So even if you dose them with drugs, they're not going to bring it in because they stopped their metabolisms and slowed it down so the drug doesn't affect them. So biofilms can be potentially medically significant. Like we don't want those on a chemo port. You don't want biofilm and plaque accumulating on your teeth because you're going to get cavities. I do have an interesting video here about quorum sensing and little guys that sense how many there are around them, and then they'll express certain genes. So if you have a chance, check that out. It's really cool. But that's a little bit about biofilm and how they will, you can have different communities living together to form this biofilm on your structures, whether it's your teeth or a port or a catheter, whatever it is. All right, so moving on to the prokaryote anatomy, all of these guys are going to have some basic characteristics for prokaryotes. They're all going to be unicellular, which just means one cell, instead of multicellular like our fungi and me and you. Then they're going to lack membrane-bound organelles. So if we took 1010, or if you remember from high school biology, our membrane-bound organelles are going to be our nucleus, our mitochondria. Um, those guys have a membrane going around the organelle, our endoplasmic reticulum, that is going to, keep things inside of that little organelle and keep things, the cytoplasm, the cytosol outside. So these guys, if you're a prokaryote, you are lacking those membrane-bound organelles. Another difference is in their DNA. For their DNA, we learned about how DNA is a double helix, right? And it's this helical structure. And for our DNA, if you've ever seen it in your high school biology or 1010, that DNA for us, you always see it as little linear stripes, right? It's always like this. And then we talk about mitosis, how you make a sister copy of that. And so they're going to meet the centromere, and then there it is. So these are going to be our eukaryotic DNA strands. But for the prokaryotic DNA strands, they are not going to have linear, they're going to have circular are DNA strands. And so they have one big circle that's got most of their DNA for most of their essential stuff in there. And then they'll have extra little circles called plasmids. And those will have extra cool things that maybe set them apart evolutionary, give them an advantage. Like maybe one of their plasmids has antibiotic resistance, methicillin resistance, tetracycline, whatever it is. They may have genes in that plasmid that allow them to be resistant to drugs. But these guys, the circular ones, all of our prokaryotes have circular DNA and our eukaryotes have linear DNA. Another big difference on these, and I always ask this test question, um, there is no membrane-bound nucleus, right? We said if it's a membrane-bound nucleus, it's a eukaryote and not a prokaryote. Our prokaryotes have their DNA. If you look, it's all this red squiggly stuff right here, right? This is all their DNA, that big long circle I was telling you about. And it's in a place called the nucleoid. Nucleoid. It's not a nucleus. It's in the center, but it's not membrane bound. So it is not a nucleus. It is called the nucleoid, this generalized region where the DNA hangs out. Other things we need to know about prokaryotes. Their shapes, they come in lots of different shapes. These are three popular shapes, but there are like 6 total shapes that we learn about in micro. But our most common ones that you would see in imagery are going to be the spherical ones, and those are called the cocci. That's these guys right here. I don't think it mentions which one this is, but for example, staph that we all have on our skin, we have staph epi on our skin, staphylococcus epi, and that is this guy. So this could be staph epi, SE for staph epi, and then another common shape would be the bacillus or the rod shape. And to me, I think like these guys look like little hot dogs or little carrots. And so E. coli that we also all have in our guts, E. coli, I cannot write with my finger. An Apple pen would be so much easier right now. E. coli. We should have just put EC. So the E. coli and Bacillus subtilis that lives in the soil, but we all have both of these. We all have Staph epi on our skin, and we all have E. coli in our guts. These are the most common shapes that you will see on imagery of prokaryotes. They're our most common bacteria that we play around with. Another shape that bacteria have that we don't play around with a whole lot. But this is a spirillum, and he's really cute looking. It looks like a little spring, right, that somebody grabbed out of your pen and pulled apart just a little bit to make it a little looser. It's the spirillum shape or spiral shape. They are going to be found. They are bacteria that is out and about. We can find these in the guts of ticks when they spread Lyme disease. This is what Lyme disease looks like out of the guts of ticks. Syphilis also has this shape to it. So definitely knowing your shapes is kind of helpful when you're looking at bacteria. So this is our coccyx shape. This is our bacillus shape, and this is the spirillum, spiral shaped. When we are trying to figure out what somebody has, one of the helpful things too is to obviously take a culture. Like if you have somebody coming in and they are ill and you don't know what they have, take a swab of it and try and grow it, culture it. And then once you culture it, then you can look at it. What shape does it have? Is it a coccyx shape? Maybe it's staph. Is it a bacillus shape? Maybe it's E. coli. What is it? So we can culture it. And another thing that we can do when we culture it, we can gram stain it. You can gram stain. So gram staining is when we look at the cell walls. When we look at the cell walls, which I think I may have left out on our imagery. Let me back up in a second. On our guys, all of our prokaryotes are going to have DNA inside that nucleoid, right? They're going to have ribosomes because we talked about central dogma, where you have DNA, and then you're going to have RNA. And then from RNA, you are going to make your amino acids, and you can't make amino acids unless you have ribosomes. So in order for us to make the proteins, for the cells to make the proteins, you got to have ribosomes. So these guys also have ribosomes, and that's all the little purple dots in here. These are all the ribosomes. So every prokaryote has DNA in the nucleoid. It's got ribosomes. It's got cytoplasm. It's going to have a cell membrane. Every bacteria has a cell membrane. Almost all the prokaryotes have cell walls, but anytime you make blanket statements, Mother Nature laughs at you. So not every bacteria has a cell wall. Most do, most do. And we will talk about those two different types of cell walls in a second when I go back to gram staining. Some of the bacteria out there will have a capsule, and usually the capsule is found in our more pathogenic ones or infectious types of prokaryotes. The capsule could allow a bacteria to be so big that the white blood cells, the macrophages that come along and try and eat it, can't eat it because the capsule makes it so big that it's hard for that thing to get it and phagocytize it into the cell. Um, Sometimes the capsule allows the bacteria to stick to surfaces, like we were just talking about biofilms on our teeth. Maybe the capsule allows it to stick to that surface of your teeth better. Pili are something that we'll talk about when we talk about bacterial reproduction and swapping genes. They'll have reproductive pili. Sometimes bacteria have flagella. Sometimes the flagella is just one flagella on one end. Sometimes they have another flagella on the other end. Sometimes they have a bunch of flagella, like a giant, I don't think you can see my hair right there, but a giant ponytail coming out the back. Sometimes they are covered in flagella, like a hairy beast. They come in all different types of varieties. So this is a hit or miss optional. The capsule is optional, but these guys, the cell walls are pretty standard. Cell membranes are definitely standard because you don't have a cell without a cell membrane keeping everything in. Ribosomes are standard. Uh, Very definitely DNA is standard on these guys. So back to the cell walls. On the cell walls, if we were trying to figure out what type of bacteria we had and we put it under a microscope and we gram stain it, not only would be able to see the shape of it, but it would also be helpful to do a gram stain because it tells us what our cell wall is made out of, what the bacterial cell wall is made out of. So on a gram positive type of bacteria, these guys will take in, it's a whole process. You're going to do crystal violet, then you're going to take in iodine to stick the crystal violet into the peptidoglycan. The iodine acts as your mordant right there to make sure that that purple crystal violet stays in there. Then, after that step, you're going to decolorize with alcohol just for a few seconds there and pull any of the crystal violet out. If it only has a little bit of peptidoglycan, like this guy, look at that. There's just a little bit of blue there for peptidoglycan. If it only has a little bit, when you put in that alcohol, it pulls the crystal violet out of that peptidoglycan, and now your peptidoglycan is naked. It doesn't have any stain in it. And then you have to do your fourth one. It's your secondary stain, and it's called safranin, and it's this beautiful kind of pinkish red stain. And that will stick in for your gram negatives. For your gram negatives. For your gram positives, you have so much peptidoglycan on these guys. Look how much all this blue is peptidoglycan. So these are gram positives, and they stain purple. This is a gram positive, purple. And then our gram negatives, we said that when we do the alcohol step, it pulls the crystal violet out of the peptidoglycan. They hardly have any peptidoglycan. So whatever they had there, that crystal violet gets pulled out with the alcohol, and then we have to stain it again with the safranin. And this is a gram negative. If we had a a gram-positive bacteria, that crystal violet's there. The alcohol comes along, tries to pull it out, but there's still so much embedded in the peptidoglycan that it stays purple. And when you stain again with the safranin, it just doesn't show up because purple's darker than the safranin. So your gram-positives will always stain purple if you're doing it correctly, and your gram-negatives should always stain that pinky safranin color. So on a test, if I'm asking you who's gram-positive and who's gram-negative, I need you to know that gram-positives have a lot of peptidoglycan. They have a lot of peptidoglycan. Look at all that blue for gram positives, and they stain purple. They stain purple. Um, on the gram negatives, very little peptidoglycan. On our gram negatives, not a lot. You can see just a little bit of blue there. And they will stain pink. They stain pink for our gram negatives. Pink. My dog is snoring so loud right now. All right, and so that's our bacteria. We can stain their walls and see if they stain purple or pink with a gram stain. So it's a differential kind of stain. On archaea, they don't use peptidoglycan in their cell walls. They use pseudo fake peptidoglycan. So it's kind of similar, but its chemical structure is slightly different. So it is called pseudopeptidoglycan. So that's what's in their cell walls. And we can use this as a diagnostic tool to figure out, do they have a staph infection? Or do they have an E. coli outbreak? Like what's going on with this person? Gram staining is something that we can do with the cultures that we swab or take from people. How do the bacteria, the prokaryotes, the archaea, swap genetic information? How do they reproduce and get new, fresh genes in their gene pool? So there's three different ways. And with each of these different ways, I kind of have like a keyword for them that I want y'all to like associate with the three different types. So we've got transformation. Transformation is where we're going to say this guy is E. coli. This is E. coli. They're all E. coli. You are E. coli. And the E. coli, in this case, it sees something in the environment. It says, ooh, that looks tasty. And it phagocytizes it, eats it, brings it inside, and keeps, look, if you see it, you can make out that little green piece there. It keeps that genetic information from the stuff that was out in its environment. It just absorbs it, keeps it, incorporates it into its genome, and now it's a part of it forever. So that's transformation. It takes stuff from the outside, genetic material from the outside of itself, brings it in and incorporates it into its genome. That's transformation. So it's phagocytosis of stuff on the outside. Then we have transduction. Transduction is going to use this guy. This is our most important keyword for transduction: viruses. Viruses. When a virus attaches onto a cell, in this case, this would be a bacteriophage because this is an E. coli, which is a type of bacteria and that type of virus that only likes bacteria called bacteriophages. So when that virus latches onto that host cell and injects its genome in there and it gets incorporated, that is called transduction. And this is happening to prokaryotes all the time, and it's happening to us because if anybody knows, there are certain viral diseases that we can get as humans that stay with us our whole life. Stay with us our whole life. So HIV, herpes. Sometimes when people get chicken pox, right, it shows up later as shingles, and we didn't know that it did that until the last couple of decades. We figured that out. But initially, when we learned about chickenpox, we didn't know it did that. So we would have things called chickenpox parties because we thought it's better for you to have chickenpox when you're little. So we exposed everybody to chickenpox, not knowing that it's going to show up later as shingles. Because it's doing this, it's undergoing transduction and it lurks in your neural column on your dorsal root ganglia. And that's when it shows up in patches. It just lines itself up with the dorsal root ganglia that it's been lurking in. And then later, it pops up as shingles. So they do have vaccines against this now. We have vaccines against chickenpox, and we also have vaccines to keep shingles from happening, to keep it dormant so this doesn't happen. But anyway, back to this type. It's called transduction. And the keyword I want you to know about it is this guy is the viruses. No, if you see a descriptor of this describing it with viruses that are infecting the bacteria and that genome gets incorporated in, it's transduction. And then our very last type of prokaryote reproduction is conjugation, which is basically bacterial sex. It's bacterial sex. They are going to see another compatible same species bacteria next to them And they're going to take that reproductive pili that we saw in that image before. This is this little reproductive pili, and it latches on to that bacteria next to it and it pulls it in really close. And then once it's got a hold of that bacteria, it can swap genetic material through that reproductive pili. It's so cool. So say this was, we're going to say this is, it's not the right shape for staph, but I'm going to pretend that it's a staph bacteria. And this is a genome, a plasmid that has methicillin resistance, MR, methicillin resistance. So it has the genetics to be resistant to methicillin, which is an antibiotic. And normally methicillin would take care of staph, but this particular staph has methicillin resistance and it's latched onto another bacteria next to it that's just like it, and it's going to pass on the methicillin resistance to it too. So now everybody has methicillin resistance. This is conjugation. This is bacterial reproduction or bacterial sex. So please know these three different types. Transformation, we are taking stuff from the environment and incorporating it into our genome. Transduction is going to have a virus associated with it. A virus latches onto the host cell, injects its genome in, and it gets incorporated. Conjugation is using a reproductive pili to latch onto a neighbor, bring it close, and swap genetic material with that little plasmid. So it's going to peel off this little plasmid right here. It's going to make a little notch in it, and it's going to peel away our double helix. And half of it's going to go across that reproductive pili to the neighbor, and then they will have their own plasmid now that they now have methicillin resistant genes inside them, which is cool in a science way, but not so great if it's if it's antibiotic resistance. So please know the three different types of prokaryote reproduction. When we talk about other considerations in here, absolutely understanding the basics of infections, what prokaryotes are, what bacteria are, what archaea are helpful, how they're spread. We started off talking about prokaryotes with good hygiene, and that child's handprint, and make sure we are washing our hands because our hands get really gross. You know what also gets really gross? The things you touch every day and you don't think about washing. This is gross. This is gross. My cell phone is nasty. I try and wash it, but I have cultured Staph Epi and Staph aureus off of my cell phone before. So just a little tip from me to you. Wash your surfaces. Those are good. All the things you touch. I can ask a question. If I was in a face-to-face class, you'd be able to answer me, but I don't know if you know how the Black Plague was spread. I hope you do. But it turns out it was the fleas that had the bacteria Yersinia pestis. I'm sure it's in here somewhere in the description. There it is, Yersinia pestis. And so those bacteria lived in the guts of the fleas, and the fleas lived on what? The rats, right? They lived on the rats. And then who was great at mousing? Cats. Cats are, right? So cats are really great at keeping the rodents under control. But at the time, there was witch hunts going on, and we did not believe in cats. We thought they were bad, and associated with witchcraft and devilry. So we did not have as the cats that we do now. Um, to control the rats. And so the rats are going willy-nilly all through the city, carrying these fleas that had Yersinia pestis in them. And then the people would get bitten by the fleas on the rats, and then they would get bubonic plague, which killed a massive portion of those people in Europe. And you think, oh, it's done. It's one and done. We're fine. It's still around. It still happens. The bubonic plague is named the bubonic plague because when you get it, your lymph nodes get swollen. If anybody's ever been to the doctor, right, you know that they come along and they start feeling these guys, and they'll feel for some of your lymph nodes. You got at least two of them right here on either side of your neck. But you have them all over your body. But if they're feeling for them and they're swollen, that usually means like, why are they inflamed? Why are they swollen? Those guys are helping with your immune system. So swollen lymph nodes are called buboes, and that's why the bubonic plague gets its name. So we'll have the bubonic plague, we have pneumonic plague, which is in the lungs. But this is just one example, one image of it on the bubonic plague. And you will still see this stuff pop up. Years ago, it was Madagascar had some plague come through. So it still crops up, it's still hanging around. This stuff can live in the soil and then it gets picked up by the rats and then it spreads. So this is just a little background. This right here is a hyperlink if you wanted to click it. It talks about the origins and how it was passed on with the plague. Typhoid fever and typhoid Mary. If you read this, this is a really good article on it. But typhoid Mary was, I want to say, Irish immigrant. And so what she did back in the day, women did not have a whole lot of career opportunities, shall we say. There were very limited things that you could do to earn a wage as a woman back in the day. And so one of the things that you could do was cook. You could be a cook. And this was Mary. Um, and so I think it was, I can't remember Mary's last name originally. She ended up changing it to Mary Brown later. But Mary was a chef. She was a cook and she was a good cook, and she would be hired on by these families as their private chef. But unfortunately, Mary's families kept coming up sick. And so the investigator was looking, and the only common denominator he could find between all these sick families and these sick kids and people were dying was the chef. And this chef named Mary was the family chef. And that's the only reason that they could figure out how these families were getting typhoid fever. But Mary was fine. She wasn't showing any symptoms. She was totally asymptomatic. And she's like, "Why are you coming after me? That's not fair. I don't have this. You can see that I don't have this. You're just persecuting me because I'm an immigrant." And she had a very valid point because there was absolutely persecution against immigrants back in the day. And there still is today, just different immigrants. But anyway, Mary got arrested and she finally got let go because she's like, "You can see I'm not sick. I am not sick." And so they let her go, but they said, "Okay, Mary, you just can't cook. Just don't cook for anybody anymore." And she was like, "Yep, Scout's on her. I won't do it." But again, she doesn't have any other profession to do. This is her life's work is cooking. So she changes her name and she starts cooking for another family. And that family also got sick later. And so it turns out that Mary gets arrested again and she doesn't get let go. She lives out the rest of her life in an asylum type situation. And upon her death, she was autopsied and she did have typhoid fever. She had the bacteria, the typhus bacteria, in her gallbladder. And so she, every time she was cooking for these families and she didn't wash her hands after she went to the bathroom, she was spreading typhoid fever to every family that she cooked for. Unknowingly, because she was asymptomatic, she wasn't showing any symptoms, but she loved to make, I think it was a peach cobbler dish with like ice cream. And so every time she made that dish, she had the potential to pass on typhoid fever. So that is a story about Typhoid Mary. If you've ever heard that expression, that's where it comes from. And then, of course, we have all sorts of other foodborne illnesses. We hear about them almost daily in the news. But we have some that have hit us lately in our local area, in the Atlanta area with Whitewater. It had an E. coli breakout. And that specific type of E. coli causes the red blood cells to rupture, and those clog up your kidneys, and then your kidneys shut down, and that shuts down everything else in your body. It's called HUC, hemorrhagic uremia, and it's not great. It's a certain strain of E. coli that can do that. Not every strain, because most of us have E. coli in our gut, and we're totally fine. It is living there just fine. But there are certain strains of E. coli that can be deadly, and that is one of them. And a child had gone to the bathroom at Whitewater. They did not have the proper chemical mixture in there to kill off the bacteria, and several other kids got sick too that day. Other foodborne illnesses. Botulism is something that I used to be like, you'll never have to worry about botulism again because that's something that, you never see that anymore. You'd have to look for dented cans at the store, and that's what you would worry about with that. But sometimes it still happens. Listeria is something that I definitely still hear about and see in the news a lot. Sometimes you'll see listeria infections with sandwich meat. Sometimes it's unpasteurized cheeses. Could be listeria, could also be salmonella. So there are lots of different foodborne illnesses that you need to be aware of and listen out for. And if you believe that you have purchased something that is getting recalled, just toss that stuff out because it's not worth it. I was telling y'all about the plasmid, and when we were talking about conjugation and bacterial sex, where they use the reproductive pili and splice out their genes and give it to somebody else, this is exactly what I was talking about. So MRSA is the methicillin resistant type of Staph aureus, but there's also vancomycin, which is another type of antibiotic resistant. Um, and then there is tuberculosis, which is currently present day in 2025 having an outbreak in I want to say Oklahoma. Midwest is having a TB outbreak. And this one, the MDR stands for multi-drug resistant, multi-drug resistant. So TB is something that is spread through the air particles from an infected person that spreads to the next person. And once you get infection, I think it takes 10 to 14 days maybe to show up for that. I'm a little fuzzy on how long symptoms show up for this, but I want to say it takes up to 2 weeks for you to show signs and symptoms of it. And it can take several months of drug treatment to try and get this thing under control and out of your body. So bacterial resistance is a real thing. It is a real problem. And if you are sick and are are you getting, are prescribed bacteria antibiotics, please take your full round. Take your full round. Don't take like half the pills and you feel better and you just leave the rest of the pills in the bottle. Finish off your antibiotics because otherwise, the ones that are left behind are resistant to those drugs. And remember how we talked about conjugation, they can pass on that resistance to their friends. So please, if you get antibiotics, take the full dose of antibiotics and kill off all of those little bad guys so they don't spread resistance to other ones. Other uses for prokaryotes. We're talking about all the bad things that could happen with prokaryotes, but there's so many good things too. If you love cheese, I love some cheese. Absolutely, prokaryotes. If you like any sort of fermented food, if you like any of this stuff here, cheeses, yogurts, all of that stuff is prokaryotes in action. If you like kimchi, pickled anything, all of that prokaryotes. Um, Alcoholic beverages, all of that, prokaryotes. Other things prokaryotes are really good for is bioremediation. So bioremediation is when there's some sort of environmental issue. In this case, we have an oil spill in this image, and you are using living organisms, the bacteria, to try and remediate the situation. And in this case, we can use some bacteria. Remember how I talked y'all about how bacteria have lots of different crazy types of metabolisms? Because they were here first and they have evolved all sorts of really neat and interesting ways to metabolize all sorts of stuff in their methods of cellular respiration. Well, some of these guys, some of them can take the oil and break the oil and metabolize it as part of their cellular respiration. So we can use some of these guys to help bioremediate. So when we had this one's talking about Exxon Valdez, which is one that I grew up with as a child, seeing and experiencing. Deepwater Horizon is another one that happened a little more recently where we had oil spills. And in that case, we sprayed lots of, um, what's my word? I'm blanking on it. But it acts like emulsifiers. That's the word I was looking for. We sprayed emulsifiers out there that act like Dawn dish soap, and they take the heavy oil and the emulsifiers break it up into smaller clumps. And then we spray the prokaryotes on there. And now that we've got smaller clumps and it's not just one big mass, it's easier for the prokaryotes to get in there and try and, gnaw away at these big giant globs of oil and try and break it down and metabolize it a little bit. Does it work all the time 100%? No, of course not. But it's one way that we can try and help out and try to remediate the damage that we have done as humans to our environment. Remember, I was telling y'all about we've only talked about the bad guys, but there's so much good things. There are lots of good guys in here. Most of our bacteria are going to have a symbiotic relationship with us. So it's a win for us and it's a win for the bacteria. And this is you can see all the whole list, all of these different major groups of prokaryotes. These are so many different groups here. Um, it's a win-win for them. They have a place to live. They have food source. They have shelter. They are happy. We are benefiting from having them here. Oftentimes, the stuff in our gut biome helps us break down our food. It helps us metabolize different types of vitamins, like our vitamin K, for example, is metabolized in the gut, and that helps us bring that in. And vitamin K helps us with blood clotting, which is something that we all need. So most of these guys are living in a symbiotic. It's a win-win for both of us. Other types, it's just commensalism. They are hanging out, vibing, doing their thing, and the host doesn't care. Like it doesn't affect the host either way. The host is just neutral, and it's a win for the bacteria that are living there. So we have microbiota, our microbiome all over us, and you can see the different places on the body where different things like to live. Um, on the outside of us, we've got bacteria, we've got archaea, we have fungal, we have yeast for sure, all around us. So we are inside, outside, we are a whole biome. It's not just us. All these little guys are living happy with us. So that's gonna leave us there with prokaryotes, and then I'll start another video on the rest of this, and hopefully it won't be near as long. I could talk about prokaryotes for a really long time. They are really fascinating. All right, bye guys.